Okay, so large intestine. The large intestine extends basically from where the ileum attaches at the ileocecal sphincter. Okay, remember, ileum and the cecum is this first part of the large intestine that drops below that connection point, all the way to the anal canal. So that is large intestine. Okay. So we've got four primary regions. The cecum, which is this kind of lower hangy part here that's below that connection. The colon, which is actually most of this. The rectum, which is just this little last part here before you get to that anal canal. Movement through the large intestine takes 18 to 24 hours. Okay, small intestine, we're talking about a maximum of 3 to 5 hours. With large intestine, it's a lot longer. Now remember something though, the small intestine does a lot of moving. You've got the peristalsis and you've got the mixing motions and you've got all of that going on. Your large intestine has about 3 to 4 mass movement events, and that's it, a day. So its movements by comparison to my small intestine are really, really slow. Okay. While in the large intestine, that chyme, that processed product that we've gotten out of our small intestine is eventually converted into fecal material or poop. Okay. Water and salts are absorbed here. I'm trying to get as much water and as much salt as I can out of the product that has come out of my small intestine. Okay, so it's getting drier and drier and drier. There is a lot of mucus secreted here, a lot. Why? Because that stuff that I am drying out, I don't want to poop powder. I need to have a way to kind of make it stick together so that I get that semi-solid mass. And the way that I do that is I've got all of this sticky mucus along the lining. So as it's going by and I'm absorbing water and I'm absorbing salt, it's also kind of getting stuck together with the mucus. Microorganisms exist in your large intestine. Now you might be going, okay, why? Well, there are things that our body cannot make. And the fact that our body can't make them means that if we don't get it in our diet, we kind of get in trouble. So the bacteria that exist in our large intestine are benefiting in the fact that they get to eat the chyme. They get to eat off the chyme. They get nutrients from that chyme material. And their waste product is a vitamin. So they excrete this waste product and we get the waste product that is a vitamin that we can actually use. So they're getting the benefit of being able to feed off of our fecal material, and we're getting the benefit of their waste material being an actual vitamin that we can take advantage of and we can use. The colon itself actually stores feces until we can defecate. It actually just kind of houses it until we can go to the bathroom. About 1,500 mils will enter your large intestine. Of course, this is average. If you're eating more, if you're eating less, that can change. But on average, about 1,500 mils enter the large intestine. But toward the end of that, the waste that's actually defecated out of the body is only going to be about 80 to 150 mils, meaning that we are reabsorbing the water and the salts out of this by about 90%. We're not just letting that go if we can help it. Most of the time, when people get diarrhea, just as an example, we tend to get dehydrated because we tend to keep a lot of the water out of our chyme that's becoming fecal material. Okay, so parts and pieces. So looking at the large intestine, we've got this ileum, small intestine coming in. There's the ileocecal valve, which is basically the entrance point for this. It's also a sphincter muscle. We've got the cecum that's coming down here. Here's the appendix, if you've ever wondered where that is. Coming up this direction, if I'm taking this material to exit, it would be coming up here, right? So this is the ascending colon. goes across, transverse colon, goes down, the descending colon. And then if you look at this, if you were to flip it around, it would look like an S. This is the sigmoid colon to the rectum and the anal canal. Now, do you see this strip here? This is called the tenai coli. This is where the muscles can kind of pull and move against, okay? Each one of these lumps that you see is called a haustra, okay? These kind of globules are called epiplatic appendages. We honestly have no clue what they're there for. They exist, that's what we know. As far as the muscular layer, remember we've got the mucosa, the submucosa, the serosa, and in the middle we've got that muscularis externa. We have the two layers, same two layers as everything except the stomach. The circular layer that does the kind of squeezing, and then those longitudinal layers that actually do the pushing. So you've got circular and longitudinal muscle. We've got crypts. Basically, we've got these tubular glands, kind of like what you saw in the stomach and the intestine, with that same kind of dipping structure to them, okay? Except in the large intestine, we call them crypts, okay? So your colon, the largest or longest part of this, is about 1.5 to 1.8 meters long. The cecum, that little section that's right below the ileum entrance, is only about 6 centimeters, okay? It's not very long. So the ascending colon goes from the cecum to this right here, the right colic flexor. Transverse goes from the right colic flexor to the left colic flexor, and you can see it flexes there, it bends, okay? The descending starts at the left colic flexor and goes until it hits the sigmoid colon, where it kind of does that S formation. And then the sigmoid colon is that S-shaped tube that extends from the pelvis to the rectum. The rectum itself is a straight muscular tube. It ends at the it begins at the end of the sigmoid colon, and it ends at the anus. The anus is only about the last 2 to 3 centimeters of the digestive tract. It's not a huge long, I mean, it's just the very, very tail end of it. So as I said previously, mucus is going to be the primary secretory product that you're going to be getting out of your large intestine, okay? It's going to lubricate. It's going to help the fecal matter stick together. The other thing is, as you increase the irritation, let's say that there isn't enough mucus and you start rubbing the sides and it irritates the lining of that large intestine, as that happens, your parasympathetic nervous system is actually going to stimulate the lining of the large intestine to make more mucus, because obviously it's getting damaged, okay? Feces, it's water, some solid substances. About 30% of the dry weight of feces is actually microorganisms, so bacteria and the like. But again, remember I said the vitamins? Vitamin K is the waste product we get from them that we take advantage of. Vitamin K is used in blood clotting, so it's really, really important. And some of it is the sloughed off epithelial cells that were lining the small intestine and the large intestine. Flatus is gas production, okay? We talk about flatulence, okay? Now, why does this happen? It depends on a couple of things. One, what is the bacterial population in your large intestine? Because sometimes they're actually responsible for producing gas, and some bacteria is more likely to produce gas than other bacteria. The other thing is, what kind of food are you consuming? Because some food tends to give you gas worse than other food products. Just kind of depends. So yeah. Okay. So unlike the small intestine, the large intestine doesn't really do mixing, okay? Instead, it does these mass movements where we start to contract and push at the very beginning at the cecum and just go all the way across the whole thing to the anus. These are mass movements, okay? From beginning to end, one squeeze, okay? One mass movement, one squeeze, okay? So the mass movement contracts over a longer distance than the peristaltic, and it propels the colon contents a considerable distance toward the anus. Now, like I said, when it comes to movement, your large intestine doesn't move that much. That mass movement only happens about 3 to 4 times a day. It isn't constantly 3-4 times a day, you're done, okay? Defecation, as I stated before, is the elimination of feces. You've got reflexes that control 2 sphincters. You have reflexes that control one of 2 sphincters. You've got an internal sphincter inside of the anus that is smooth muscle. When feces is pressed against it, it automatically relaxes. And you kind of get I have to go to the bathroom. But there's an external sphincter here that is skeletal muscle, that is voluntary. You control this muscle, okay? So if you get that feeling, you squeeze this muscle, you don't automatically go to the bathroom. And it's a sensation that we have to learn. That's why babies have to be potty trained, because it's not automatic that you know that means, oh yeah, I have to go to the bathroom. You have to kind of learn what the sensation means. But that external anal sphincter, the skeletal muscle one, is voluntarily controlled, and it regulates the movement of feces. You don't just automatically go to the bathroom because of a signal from the smooth muscle sphincter. You clench and you get to a bathroom until you can release and then go. Okay, so let's talk digestion, absorption, and transport. So digestion is the breakdown of organic molecules into their component parts. Absorption and transport is the means by which molecules are moved out of the tubing of my digestive tract and distributed throughout my body. Transport occurs via 2 main routes. So water, ions, anything that's water soluble, anything that will dissolve in water, okay? These products of digestion are transported to the liver by the hepatic portal system, where they're processed, okay? Because they go through the blood. However, products of lipid digestion, they get transported via the lymphatic system. Like I said, you're taking something relatively small, making it into something big and bulky. It won't just go into those capillary beds. It can't. So it's almost like it takes a more indirect route. It does eventually get to the cardiovascular system. And you can see in chapter 22 how we absorb in one place and then it goes through the filtration of all of the lymphatic nodules. And eventually that fluid always goes back into general circulation in the thorax. But it's a very roundabout way of getting there. Still gets there, just a very roundabout way of doing it. So let's talk carbs first. Carbs consist of starches, glycogen, sucrose, lactose, glucose, and fructose. These are monosaccharides, disaccharides, polysaccharides. Polysaccharides are going to be broken down eventually into monosaccharides by a whole host of different enzymes, which we're going to talk about in a minute. The monosaccharides are the ones that are absorbed by the intestinal epithelial tissue, and we do this via symport. So we're moving 2 things in the same direction, but we're using the movement of one thing to kind of charge up my channel, and then moving the second thing using that charge that we've gotten. Now, what are we moving? Sodium. Sodium is the easiest thing to move into the cell because we've got a ton of it outside of the cell. The other way that we can move these other than symport is by facilitated diffusion. If we've got a channel that's kind of like a revolving door at a bank, it can help us to move things just going with the concentration gradient. Monosaccharides, they are carried to the liver, And anything that is not glucose gets converted into glucose in those hepatocytes. Remember I told you there was conversion of nutrients or changing of nutrients? This is one of those. So if I've got, let's say, galactose instead of glucose, galactose will get converted into glucose. If I've got fructose instead of glucose, it'll be converted into glucose. Okay? Glucose is what is transported to the cells that are requiring energy. This is my fuel source. This is how I make ATP. I've got glucose to do that. Okay? Insulin influences the rate of glucose transport. We know that. Insulin, if we've got a whole ton of glucose, it starts going into the cells just to get out of the bloodstream. So, looking at the path, we've got the mouth, the stomach, the duodenum and the pancreas, and then the lining of the small intestine. Okay? So, in the mouth, the enzyme that we have is salivary amylase. This is going to take polysaccharides, starch, and turn it into disaccharides. So you're talking about fructose, maltose, lactose. Okay? We're breaking down the starch into these simpler things. In the stomach, there is no carbohydrate digestion. Notice there's nothing here. Okay? And that's because we don't have any enzymes that'll work in that high acid environment. In the duodenum, remember that that's the first part of the small intestine, and that is where the pancreas puts its product. The pancreas secretes pancreatic amylase. Now remember, amylase takes starches and breaks them down into simpler sugars. So anything that wasn't taken care of in the mouth is now going to be broken down into a disaccharide, at least from a polysaccharide. Okay? Now, remember, in the lining of my small intestine, I have those enzymes, and as stuff gets smeared by, they are able to digest it. So in the lining of that small intestine, I have lactase, sucrase, maltase, and isomaltase. Lactase is going to take lactose and break it down if you have it. If you're lactose intolerant, you don't make that enzyme, which causes issues. Sucrase is going to take sucrose and break it down. Maltase is going to take maltose and break it down. Isomaltase is going to take maltose and break it down. Maltose and break it down. Okay, so how do we absorb these monosaccharides once we have them? So here is my villi and my small intestine. There's my capillary bed and my lacteal. Here's the food that's going to get absorbed into this space. So here's one of these cells. Remember I said symport. So here's my symporter, right? Once I get it into this cell, I've got a higher concentration here than I have over here. So this is just diffusion, really. It's facilitated diffusion. I've got, again, that revolving door, but it'll just go from the higher concentration to the lower concentration here. Now, once it goes into my villi, monosaccharides can go into the bloodstream. You can dissolve sugar in water, right? So it eventually goes into the bloodstream and goes into the liver, or goes toward the liver, so the liver can process as it needs to. Now, let's talk lipids. Lipids are fats, right? So we've got triglycerides, we've got phospholipids, steroids, fat-soluble vitamins. All of these things act as lipids. So emulsification is the transformation of a large lipid droplet into a smaller droplet. It's like taking a pie and cutting it into pieces, okay? So when I talk about emulsifying fats, I'm not talking necessarily about chemically breaking them down. I'm just saying I'm taking a big drop and making it into smaller drops, okay? Smaller droplets. Bile salts do this for us, okay? The bile that's coming from our liver and our gallbladder, they're doing that. Now, lipase. ACE means enzyme, lipid. This is a lipid enzyme. So this actually does cut up the lipid. It digests the lipids to form basically free fatty acids. So you've got fatty acid tails floating around, and glycerol, which is going to be basically, I can't think of the name. A glycerol and a fatty acid tail. Monoglycerides, there we go. The micelles. Micelles form around lipid digestion products, and they move to the epithelial cells of the small intestine. Remember, the tails of my phospholipid membrane don't have any issues with lipids going through. They don't care because the lipids hate water too, okay? But the micelle is actually what helps them to get there. The micelle is basically the product of the bile salts surrounding that little lipid molecule inside. Inside of the epithelial cells, those free fatty acids are combined with glycerol to make triglycerides. This is what's floating in our blood that we go and get blood work done for and everything else. Proteins coat the triglycerides, the phospholipids, and cholesterol, and they form something called a chylomicron. Okay, so chylomicrons are the unit, basically, that enters the lacteal in the intestinal villi. So instead of going into the bloodstream, I'm going into the lacteal that's going to take me into the lymph. Okay, they get carried through that lymphatic system until they can get dropped off in the bloodstream. Again, indirect route, but eventually you end up in the same place. So looking at the parts and pieces, again, I've got the mouth, I've got the stomach, I've got the duodenum with the pancreas and the lining of my small intestine. So in the mouth, you do have a lipid enzyme. It's called lingual lipase. Lingua means tongue. So the lingual lipase is going to be an enzyme in your mouth that can help to digest fats. Does it work real well? Not really. In your stomach, you do have an enzyme called gastric lipase. Remember, gastro is stomach. And then coming from the pancreas into the duodenum, you've got lipase and you've got esterase. Again, two enzymes that help to kind of further that breakdown. The lining of the small intestine doesn't really have any extra enzymes. By the time it's gotten past the duodenum pancreas junction, you should have monoglycerides, again, glycerol with one fatty acid hanging off it, and then those free fatty acid, those chains that are coming off. So here's my villi, here's my little capillary network with my lacteal in the middle, here are the cells. So if this was the food, this would be the food. The bile salts have surrounded this globule of fat, fatty acids, monoglycerides. This is the micelle, okay? So remember I called it a micelle? This is that, okay? Mycele is another way that I've heard people say it, but I learned it micelle, so that's the way I say it. Anyway, this gets transported into this cell, okay? And I've got the triglycerides. Like I said, the tails in this plasma membrane don't have any issue with this coming through because it doesn't like water either. So the triglycerides in here, well, the monoglycerides and the free fatty acids get converted into triglycerides, get a protein coat around them. This is the chylomicron, okay? So the chylomicron gets put into this space here, can't fit into the capillary, so it has to go into the lacteal. The lacteal will now basically carry this through the lymphatic vessels until eventually it drops it off in the bloodstream. So, triglycerides. These are stored in adipose. They're converted into other molecules, and they can be used as energy. Lipoproteins are a combination of a lipid and a protein, okay? Basically being moved around. It's a combo of a lipid and a protein because I'm trying to move this lipid through the watery environment of the blood. So the lipoproteins that we have traveling in our blood include the chylomicrons. Chylomicrons are only about 1% protein. Mainly they're made out of lipids. We've got very low density lipoproteins, 92% lipid, only 8% protein. Low density lipoproteins, which are 75% lipid, 25% protein. And then we've got high density lipoproteins, which 55% lipid, 45% protein. Now you may be going, okay, I know I've heard of HDL and LDL. Yes, you have. When you get blood work done, they talk about your triglyceride level, they talk about your LDL and your HDL. And usually if you have a high HDL, they're like, yeah. If you have a low HDL, they're like, ooh. If you have a high LDL, they're like, ooh. And the reason is because of what they're doing. So low density lipoproteins, they are taking cholesterol out into your body. So I'm not getting rid of the cholesterol, I'm actually putting it inside my body somewhere, okay? It's taken into cells via receptor-mediated endocytosis, which is controlled by a negative feedback mechanism. Now, why is that bad? Cholesterol in your bloodstream, in your tissues, in your body will actually cause plaques to form, which can lead to cardiovascular disease. So it's not a good idea. And with LDL, I'm literally taking fat and cholesterol and putting it in the vessels of my body. This is not a good thing. However, HDL, this is taking cholesterol from around your body to the liver. So I'm not sending it out into the body. I'm literally bringing it to the liver. Now you're probably going, okay, for what? So the liver can use it. The liver can break it down. It can make it into something different, like bile, because bile salts are actually made from cholesterol. That is one of the things that it actually is made from. Well, it is made from, not actually, it just is. So when I do that, when I have HDL carting this cholesterol to my liver, basically I'm getting rid of it. I'm not letting it sit in my tissues, causing the harm that it could by making plaques. So again, HDL being high, most doctors are real happy about that. LDL, not so much. So let's talk proteins. So we mentioned this earlier, pepsin in the stomach actually cuts the protein into polypeptide chains. We've got proteolytic enzymes from the pancreas that are also cutting and making smaller peptide chains. And then we've got peptidases, which are enzymes in the lining of the intestine or along the lining of the intestine, small intestine, that also cut them into smaller chains. What we're actually absorbing is the individual amino acids. So remember, proteins are built out of amino acids. They're just long chains of amino acids. So when I want to absorb things from a protein, I want it to be just the one amino acid. I don't want it to be chains of them. Amino acids are transported to the liver, where the amino acid can either be modified and released into the bloodstream, or released into the bloodstream. The amino acids are actively transported into the cells under the stimulation of things like growth hormone and insulin. And for us, mainly, amino acids are going to be used as building blocks. I can use those amino acids to build another protein. Technically, I can use it as an energy source, but that's kind of last resort material. If I have sugar, or I should say carbohydrates or lipids available, I will go after those before I will go after amino acids. Amino acids are more beneficial as a building block for me to make a new protein out of. So again, in the mouth, we don't have anything that digests proteins, but in the stomach, we've got pepsin. So pepsin is going to cut them. Now remember, the hydrochloric acid, all it does is it unravels it. It doesn't actually cut anything. Pepsin does. So we go from having full proteins to having these long chains of polypeptides. As we get to the duodenum with the pancreas, we've got trypsin, chymotrypsin, and carboxypeptidase. These enzymes are going to take these long chains of amino acids, and they're actually going to cut them into smaller chains. So you end up with very small peptide chains. Again, in the lining of the small intestine, the ones that are along the walls of the small intestine, I have aminopeptidase and peptidase. By the time that I get to this point, I'm going to be left with basically three forms of protein. I'm either going to have individual amino acids, I'm going to have dipeptides, so 2 amino acids stuck together, or I'm going to have tripeptides, 3 amino acids stuck together. But I'm not going to have anything bigger than that. I shouldn't have anything bigger than that. So, again, here's my villi, there's my blood vessel, here's the food. The dipeptides, tripeptides, or amino acids get put into my cell here, okay? So I'm absorbing them into the cell. Inside the cell, if it is not an individual amino acid, it gets made into an individual amino acids. So if I've got a tripeptide coming in, I'm going to break those 3 amino acids apart so that they're individual. If I have a dipeptide coming in, I'm going to break them apart so I have 2 amino acids instead of a dipeptide. Once it's in there, I move it into my villi where I've got my little capillary bed, and it's going to go into the capillary because it's water soluble, and eventually it will work its way to the liver. So, water and ions. Approximately 9 liters of water enter the digestive tract each day. 92% is absorbed in the small intestine. About 6 to 7% is absorbed in the large. Water movement is concentration dependent because when we talk about water moving, we talk about osmosis. Now remember, osmosis is the movement from a high concentration of water to a low concentration of water. So if you're dehydrated, like severely dehydrated, and you drink water, it'll go into your body like nobody's business. However, it won't work the opposite way if your water balance is that off. So things that come in with this, you've got sodium and potassium and calcium and magnesium, phosphate. They're actively transporting in. Chloride will move passively through the duodenum and the jejunum, but in the ileum, that last section of your small intestine, it actually does have to be actively transported. Calcium is actively transported, but vitamin D is required for transport. Transport is hormonally controlled. We talked about this in AP1. We talked about parathyroid hormone. We talked about calcitonin in the calcium balance in the skeletal system. We also talked about those 2 hormones when we were talking about the endocrine system and how they help us with our calcium balance. So, the effects of aging on the digestive system. Okay, so remember that the inner lining is the mucus layer, right? We've got connective tissue, we've got those muscle layers in there, and we're supposed to be secreting a whole heck of a lot of mucus so that it's very well lubricated, right? It tends to decrease with aging. If you've ever known somebody who's older, usually as they get older, they start to eat a lot more fiber. And one of the reasons that they do that is because of this. It actually causes issues with the movement. It causes issues with things sitting in there for so long that it's losing too much water and it's almost causing constipation. So by adding fiber, something that we cannot digest at all, it actually adds to the bulk and makes it kind of hold more water. So it stays more, lack of a better way to say it, moist, so that it's easier to get rid of. The changes that I just mentioned also cause issues with a person being more open to infections. If you're rubbing something and there's no lubrication next to it, you're going to cause damage. That increases the likelihood of having an infection. It also increases the likelihood of having infectious agents getting into the body because you're damaging the lining of the intestine. So it's an issue. Everything goes downhill as we age.